Assalamu alaikum respected doctors, welcome to my YouTube channel MBBS and BDS lectures. Today is the 8th video of rapid revision course of periodontics, which is very important for NLE, MDS, NBD as well as for BDS students. In this video we will discuss about dental plaque. In content we will discuss about in dental plaque, definition of dental plaque, then classification of dental plaque. Next is the microorganism which are responsible, which are present in the dental plaque. Next is the three steps of dental plaque formation, and in the last there are two hypotheses, specific and non-specific hypothesis, which was come in NLE previous exam. First we start from definition of dental plaque by WHO in 1961. How WHO define the dental plaque? It is a specific but highly variable structural entity resulting from sequential colonization of microorganism on tooth surface. Mean in sequence microorganism come and adhere on the tooth surface and make the colony, and it may be on the tooth surface or on restoration or other parts of oral cavity. And dental plaque is composed of salivary components like mucin, desquamated epithelial cells, debris and microorganism, and these all microorganism debris all embedded in a extracellular gelatinous matrix. This is the dental plaque definition according to WHO. Next is the Bowen definition in 1976. How Bowen defined dental plaque? Dental plaque can be defined as soft deposit that form the biofilm adhering to the tooth surface or other hard surfaces in oral cavity, including removable and fixed restoration. Mean dental plaque is a soft deposit that form the biofilm, which is pellicle, which are adhere to the tooth surface or other hard surfaces in oral cavity, including removable and fixed restoration. Now from these two definitions we get a point. Dental plaque is not only formed on tooth surface, it formed other than tooth surface like fixed or removable restoration or processes. Now come to the dental plaque classification. We classify the dental plaque on two bases. Number one, by location on teeth where plaque is located. Second, by pathogenic effect, mean which type of pathologies are mean dental plaque cause which pathologic effect. First classification by location on teeth. There are we divide the dental plaque into two. Number one is the supragingival plaque and second is the subgingival plaque. Supragingival plaque is present above the gingiva and subgingival plaque is present below the gingiva. Then supragingival plaque we divided into two types, and subgingival plaques we further divided into three types. Supragingival plaques were divided into two types is number one coronal plaque and second is the marginal plaque. If a dental plaque present on the crown of tooth above the gingiva, it is known as coronal plaque. If supragingival plaque present on the cervical margin of the tooth, this is known as marginal plaque. Now come to the subgingival plaque types. There are three types of subgingival plaque. Number one is attached to the tooth below the gingiva. Second is unattached plaque, and third is the attached to epithelium. Now in below diagram, look at in this diagram, there is a tooth attached plaque. Next is the unattached plaque below, and next is the epithelial associated plaque. This plaque is only associated with the sulcular epithelium. Next, classification by pathogenic effect, mean which pathogenic effect plaque produce. If plaque cause caries, mean if plaque responsible for caries, this is known as carcinogenic plaque. If plaque cause periodontal disease, this is the periodontal disease producing plaque. If plaque deposit and it calcified, it become calculus, and because this calculus is formed due to plaque, this is known as calculogenic plaque, because this calculus is formed due to plaque. This is the classification of dental plaque. Now come to the detail of these types. First is the supragingival plaque. Now come to the detail of these types. First is the supragingival plaque. In supragingival plaque, those bacteria which present on the tooth surface and those bacteria which form the outer surface of the plaque. Those bacteria which form the inner surface, which present on the tooth surface, are gram positive, and bacteria which present on the outer surface of mature plaque is a gram negative. Next, on the tooth surface, mean on the inner surface of supragingival plaque, there with gram positive cocci or cocci, there is a short rods. But on the outer surface with gram negative rods and filaments, there is a spirochetes which present on the outer surface. Now, gram positive cocci and short cocci or cocci and short rods are at tooth surface, and gram negative rods and filaments, spirochetes on outer surface of mature plaque mass. Now, supragingival marginal plaque cause gingivitis because there is a marginal gingiva inflammation due to this plaque, and the supragingival plaque mean coronal plaque calcified and form calculus, and this calculus will be responsible for root caries. Now come to the subgingival plaque. Subgingival plaque, in subgingival plaque there is a in subgingival plaque, if in coronal part there is a predominant filaments bacteria. Coronal part dominated by filaments, and in apical part of subgingival plaque there is a spirochetes, cocci and rods. Tooth now the tooth associated in subgingival, there is a tooth associated and epithelium associated, mean gingival associated. The tooth associated subgingival plaque responsible for calculus and root caries, and tissue associated subgingival plaque responsible for different form of periodontitis, mean inflammation on the whole periodontium, and there is a loss of alveolar bone. Now here in this diagram, the tooth on the right side is a tooth associated cervical plaque. There is a filaments, gram positive cocci and rods, and tissue associated sublingual there is a gram negative rods, cocci and filament rods and spirochetes. And the here in this diagram, the apical area of plaque mass, means subgingival plaque, is limited by a layer of leukocyte. This is about supragingival and subgingival plaque. Now come to the now in subgingival plaque, coronally there is a filaments, and in apical area there is a spirochetes or spirochetes, cocci or cocci and rods. Now these are very important for MCQ's point of view. You should remember in supragingival plaque, on tooth surface there is a gram positive and short rods, on outer surface there is a gram negative rods, filaments and spirochetes. In subgingival plaque, now next the marginal plaque, supragingival marginal plaque responsible for gingivitis, and coronal plaque is responsible for calculus. And subgingival, the coronal part of subgingival plaque, there is a filaments are dominants, and in apical parts, spirochetes, cocci, and rods are dominant. These are very important points. And the tissue-associated subgingival plaque are responsible for calculus and root caries, tooth-associated, and tissue-associated subgingival plaque is responsible for different form of periodontitis. Next, come to this diagram. In this diagram, there is a below is the in bottom there is a tooth surface. Now, this diagram show how plaque are formed, how plaque develop. Now, in bottom there is a tooth surface. On tooth surface, when we not brushing for some days, the in our saliva there is a protein which attach on our tooth surface and form a pellicle. And pellicle is a that structure to which food debris and bacteria are attached. I mean, it is a adherent site. Now on pellicle, there is a bacterial microcolonies are found. Mean bacteria come and attach on pellicle. When the first bacteria come and attach to the pellicle and form colony, this is known as primary colonizer. And after primary colonizer, the second bacteria which come and attach to the first colonizer bacteria, this is known as secondary colonizer. And between these bacteria, there is an intermicrobial matrix which consists of organic and inorganic parts. This is all about, this diagram show all about how plaque formed on tooth surface. Now, bacteria in plaque. More than 500 distinct microbial species in dental plaque. 1 gram, this is very important for MCQ's point, 1 gram plaque consists of 2 into 10 2 into 10 की power 11. Meaning 1 gram of plaque there is a 2 into 10 की power 11 bacteria. Bacteria account for now if we see the weight of the dental plaque, the most of the weight of dental plaque is found by bacteria. Bacteria account for most of the plaque weight. Non-bacterial microorganism other than bacteria microorganisms are also present in the plaque, which is mycoplasma species, yeast, protozoa and viruses. Now come to the intercellular matrix. Mean between bacteria there is an intercellular matrix. In intercellular matrix form the 20 to 30% of plaque mass. Mean if 20 to 30% of plaque mass is formed by intercellular matrix, then 70 to 80% of plaque mass is formed by bacteria. Now, this intercellular matrix is derived from saliva, gingival clavicular fluid, and bacterial products. Intercellular matrix also contain a few host cells like epithelial cell, macrophage, and leukocytes. Now, in intercellular matrix, there is a organic material and inorganic material. First, organic material. Organic materials maintain integrity of biofilm because this biofilm which is a pellicle is mainly found by proteins. Mean organic material maintain integrity of biofilm. In this organic material, polysaccharide produced by bacteria. Protein is come from gingival clavicular fluid, and glycoprotein is come from saliva, and lipids is come from debris from the from where lipid come debris from the membrane of disrupted bacterial and host cell and possibly food debris. This is the intercellular matrix, and these glycoprotein, protein, polysaccharide are responsible for pellicle formation. Now come to the inorganic component. In inorganic component, the predominantly there is a calcium and phosphorus predominant, and trace of means small amount sodium, potassium and fluoride. The source, a source per supragingival plaque. Per supragingival plaque, the source of inorganic component is saliva. Per subgingival plaque, the source of inorganic component is gingival clavicular fluid. Next, there is a fluoride which derived from external source like fluoridated toothpaste, rinses, and fluoridated drinking water. Now come to the formation of plaque. After composition, come to the formation of plaque. Now, plaque is a highly ordered and predictable ecologic succession, and there are three steps in plaque formation. Number one, first step is the formation of pellicle. Second step is the initial adhesion of bacteria to the pellicle and colonization of bacteria. Third step is the secondary colonization and plaque maturation. Now come to the first. First step is the formation of dental pellicle. Now, how dental pellicle formed? There is a selective adsorption of environmental micromolecule. There is a micromolecule which come from saliva or gingival clavicular fluid, which is protein, glycoprotein, polysaccharide. These are macromolecules which selectively adsorption aid on the tooth surface to form a pellicle. How it aid to the tooth surface? By mechanism through electrostatic forces, van der Waals forces, and hydrophobic forces. Now the pellicle. Now pellicle act as a protective barrier, provide lubrication and prevent tissue desiccation, provide a substrate to which bacteria in environment attach. Pellicle provide a substrate to which bacteria in the environment attach. Plaque does not deposit on tissue surface. Epithelial clogging disrupt bacterial population. If plaque does not deposit on tissue surface, there is a epithelial slogging, there will be disrupt bacterial population. Now after first step, formation of pellicle, next is the initial adhesion and colonization of bacteria. Now in initial adhesion and colonization of bacteria, there are 5 steps we will discuss one by one. Now in initial adhesion and colonization of bacteria and pellicle, when pellicle found, within a few hours bacteria come and attach to the pellicle. Initially, remember, gram positive bacteria come and attach to the pellicle, which is facultative microorganism which may live in mean aerobic and anaerobic environment. But in the last stages, there will be anaerobic microorganism due to deprived of oxygen environment. Remember, on pellicle, within a few hours, bacteria come, and initially it is a gram positive facultative microorganism. Now there is an initial colonizer. The first bacteria which attach to the pellicle is known as initial colonizer. When the second bacteria come and attach to the initial colonizer, this is known as secondary colonizer. Now in initial colonizer, these are very important, these name. Initial colonizer bacteria are Actinomyces viscosus and Streptococcus sanguis. Now these adhere to pellicle through adhesion. For example, A. viscosus possesses fimbriae which present in bacteria. Protein adhesion on these fimbriae specially bind to proline rich protein in the dental pellicle. The fimbriae of the bacteria attached to the proline rich protein in dental pellicle. Now in initial adhesion and colonization of bacteria, there are 5 steps. First is the transport to the tooth surface. First bacteria will transport to the tooth surface by which type of motion? Brownian motion. We already studied Brownian motion in inflammation. There the neutrophil move by Brownian motion, like in a round circle motion. Brownian motion. Next is the active bacterial movement. Third is the liquid flow through liquid. And next is the sedimentation of microorganism. This is the transport method, mean through Brownian motion, active bacterial movement, liquid flow, and sedimentation of microorganism. After transport to the surface, there is an initial adhesion. Now, the initial adhesion is the reversible stage. Initial reversible adhesion of bacterium. Now, the initial adhesion is reversible. This is important point. And the total interaction energy is the total mean how initial adhesion occur through total Gibbs energy. We are already in study Gibbs free energy. Now the MCQs. Now this point is important. The initial adhesion of bacteria occur with the dental follicle through energy interaction, which is the energy total Gibbs energy, and the transport of the tooth surface by Brownian motion, active bacterial movement, liquid flow, and sedimentation of microorganism. Third stage is the attachment. After initial adhesion, now there is a attachment. Attachment is a firm anchorage between bacterium and the surface established by specific interaction. These specific interaction are covalent, ionic, or hydrogen bond, and this is a irreversible stage. Attachment is a irreversible, and there is a firm anchorage between bacterium and surface established by specific micro interaction, and these interaction are covalent, ionic, or hydrogen bonding. Fourth stage is a colonization of the surface and biofilm formation. Now there will be colony formation on the surface and biofilm formation. The transition from early aerobic environment to highly oxygenated, oxygen deprived environment in which gram-negative anaerobic microorganism predominate. In initial stages, there is a facultative aerobic organism is predominate. In the last stage, there is a transition from aerobic environment to highly oxygenated deprived environment in which gram-negative anaerobic microorganisms are predominant. Next is a secondary colonization and plaque maturation. This is the last stage of plaque formation. Now the secondary colonizer, those bacteria which come later and attach to the primary colonizer bacteria are the Prevotilla intermedia, Prevotilla losci, and Propyromonas gingivalis, and Fusobacterium nucleatum, and Capnocytophagia, etc. These are the secondary colonizer. Per MCQs, you should remember the secondary and primary colonizer name. Now, how secondary colonization and plaque maturation occur? In secondary colonization, there is a coaggregation. Coaggregation means one organism attached to another organism. Mean the secondary colonizer bacteria come and attach to the primary colonizer bacteria and eat their product. This is the coaggregation. Ability of different species and genera of plaque microorganism to adhere to one another. This is the coaggregation. Now in secondary colonization, remember promiscuous for coaggregation occur through highly specific interaction of protein and carbohydrate molecule located on the bacterial cell surface. Mean these occur through a specific interaction of protein and carbohydrate molecule located on the bacterial cell surfaces. Now there are two forms of coaggregation. Number one is the corn cob, like corn. Second is the test tube brush. Mean these two coaggregation form when secondary colonizer attached to primary colonizer bacteria. These two form are formed. Number one is the corn cob. In corn cob structure, in which, for example, streptococci adhere to filament. Remember, in corn cob form, the streptococci adhere to filaments of B. Maturity or actinomyces species, and second in test tube brush form, structure composed of filamentous bacteria to which gram-negative rod adhere. Mean in corn cob there is a streptococcus adhere to filament, and in test tube brush structure filamentous adhere to gram-negative rods. These are the differences. In corn cob, Streptococcus adhere to filaments, and in test tube filaments adhere to the gram-negative rods. Now these names are important in the diagram. Initial colonizer are Streptococcus mutans, Orellus, Singuus, Gordoni intermedius, and Villionella, Pervula, and Actinomyces odontolithicus. And in the secondary colonizer, there is a Cypnocytophaga and C. rectus, nodatum, nucleatum, P. intermedia, P. micros, P. negrescens, and P. gingivalus, P. denticola, P. poricynthesis. These names are very, very difficult. Now come to the last part of this chapter. Hypothesis about plaque. There are two types of hypothesis. One is the non-specific plaque hypothesis, second is the specific plaque hypothesis. According to non-specific plaque hypothesis, there is no specific organism in the plaque which is responsible for disease. The whole plaque is responsible for disease. This is the non-specific plaque hypothesis. Means there is no specific microorganism, but the whole plaque is responsible for disease. In specific plaque hypothesis, this hypothesis says whole plaque is not responsible for disease causing because there is a non-bacterial part. But some specific microorganisms, some specific bacteria are responsible for specific disease condition. This is a non-specific plaque hypothesis and specific plaque hypothesis. In non-specific plaque hypothesis, non-specific and specific plaque hypothesis delineated in 1976 by Walter Losch. Non-specific hypothesis says elaboration of nauseous product by entire plaque flora. I mean entire plaque flora is responsible for, they produce a nauseous product which is responsible for disease. But according to specific plaque hypothesis, state that only certain plaque is pathogenic and its pathogenicity depend upon the presence or increase in specific microorganism. Means there is a specific microorganism which is responsible for specific condition. Now this specific microorganism which is responsible for different disease are the microorganism according to this specific plaque hypothesis. In per specific periodontal disease, there is a specific microorganism. Microorganism associated with a specific periodontal diseases. First, in normal state of periodontal health, there is a coccoid, mean gram positive organism, and filamentous form and gram negative organism. In small number on outer surface of microbial layer and few flagellated cell or spirochete. Mean in normal state of periodontal health, there is a gram positive organism, coccoid, there is a filamentous form and gram negative organism. Gram negative in small amount on the outer surface of microbial layer, and few flagellated cell or spirochetes are present. Mean in normal state of health, flagellated cell or spirochete are few. Gram negative bacteria is present in small amount and gram positive bacteria are present. Now come to the gingivitis. In gingivitis, the gram positive rods and cocci are 56% and gram negative cocci and rod are 44%. Bacterial deposit are 0.4 mm or more in thickness, and pregnancy associated gingivitis, mean for pregnancy associated gingivitis, the specific microorganism is a P. intermedia, mean Probitilla intermedia. Now come to the chronic periodontitis. In chronic periodontitis, in this transition zone, increase in number of flagellated bacteria. Mean for chronic periodontitis, flagellated bacteria are responsible. There is the increased number of flagellated bacteria. Now come to the aggressive periodontitis. There is a actinomycetes actinomycetum comitans is 90%. P. gingivalis, Echinilla corridens, C. rectus, F. nucleatum, B. capillus, U. bacterium brachy, Capnocytophaga, and spirochetes. These are the bacteria which present in the aggressive periodontitis, but the actinomycetum comitans are in 90%. And also there is a herpes viruses including Epstein-Barr virus 1 and HCMV. Next is the chronic periodontitis. Next is the necrotizing periodontal disease. For necrotizing periodontal disease, P. intermedia and spirochetes are responsible, mean these specific organisms are present in this condition. Next is the abscesses of periodontium. For in abscesses of periodontium, there is a F. nucleatum, P. intermedia, P. gingivalis, P. micros, and P. fluorescens are responsible. This is all about dental plaque in periodontics. In the next video, we will discuss about calculus. If you like my video, press the like button and share this video with your friends. If you are new to my channel, subscribe my channel by pressing the red subscribe button and also press the bell icon in order to receive my each new video notification. For watching my videos, I am very thankful to you. Thank you very much.